Welcome biologists to part 2 of Spec Point B, where we're going to take a look at the functions of the liver. We already looked at the structure in part 1, so this video we're going to focus just on the functions. There are 3 main functions we need to know about here: the production of urea, detoxification, and also glycogen storage. So urea is a really toxic compound. It's our nitrogenous waste products from the body, and it's there as a result of the breakdown of proteins, excess amino acids. The ornithine cycle is one of the 2 stages involved to form urea. The first stage is deamination, and the second stage is the ornithine cycle. And we've got detoxification, which is where the hepatocytes will remove and break down toxic substances from our diet. Now, all of these things that we're talking about here occur within the hepatocytes of the liver. Please refer back to video 1 if you want to look at more of the structure. So the first stage of urea formation is the deamination stage. So deamination is a result of the excess amino acids not being able to be stored within the body. So it's really important that we break these down. We don't want just to get rid of amino acids from the body. We want to break them down so we can use some of the useful parts of that amino acid in our other functions within the body, which I will explain in a few minutes. The second stage of formation of urea is the ornithine cycle. Now, you only need to look at this in outline only. You do not, need to do this in detail. So deamination, this is where I have amino acids, so don't forget these are in excess, and they are added to oxygen to create a keto acid and ammonia. This ammonia needs to be broken down further. It's a toxic compound and it needs to be broken down further to make urea in the ornithine cycle. So this keto acid that's produced can be used in aerobic respiration. It's used in the Krebs cycle of aerobic respiration, which we will look at when we get to respiration. This is just to remind you about what an amino acid looks like and the deamination process. So it's the amine group that's removed from the amino acid to form the ammonia in deamination. And then the second stage is where I make urea through the ornithine cycle. Now the ornithine cycle, like I said, you only need to look at this in detail. This is where I have ammonia plus carbon dioxide makes urea and water. You do need to know the word and the symbol equations for this. You do not need to know this in detail like in this image anymore, you just need to look at it in overview. So the key thing here is that this urea that is made in the hepatocytes in the liver is transported by the blood to the kidneys, where it's extracted from the blood in the process called ultrafiltration, which you'll learn about in the first part of the kidneys. The ornithine cycle requires ATP, so it is an active process. But the good thing here is that this urea is less toxic to the body, which is why we do this ornithine cycle and why we do this conversion. The next thing that we're going to look at is glycogen and glycogen storage. So this occurs within the hepatocytes in the liver. And this is basically the carbohydrate storage in animals and mammals. We store glucose as a carbohydrate storage known as glycogen. This is stored in the hepatocytes, which are the cells inside the liver. You do need to know about the structure of glycogen, which links back into biological molecules. You need to know the bonding present. So just to remind you, it's got an alpha 1 to 4 and an alpha 1 to 6 glycosidic bond. These 1 to 6 glycosidic bonds give it this highly branched structure, which also mean that it's energy dense and that I can easily hydrolyze the ends to release a glucose for aerobic respiration. It's also not going to impact upon water potential, and it's really good energy storage. The next thing we need to know about is detoxification, and the way that we look at this is detoxification of alcohol. The main alcohol present in the alcohol that is drunk is ethanol. Ethanol is converted into ethanal, so again this is all occurring in the hepatocytes of the liver. And the way that this conversion occurs is using this enzyme called ethanol dehydrogenase, which removes a hydrogen ion from the ethanol. This hydrogen ion is gained by an NAD, which is a coenzyme. This coenzyme NAD gains my hydrogen ion to become reduced NAD. This results in ethanol. Ethanol is then converted into acetate using this enzyme ethanol dehydrogenase. And it does this by gaining the hydrogen ion off my reduced NAD to form acetate. Because my reduced NAD is then lost a hydrogen ion, it reforms NAD. So the good thing about this is that NAD is recycled. Once I've got my acetate or my ethanoate, this then enters the Krebs cycle, which again is a stage of aerobic respiration, which you're going to learn about. And the whole idea here is that I'm not losing any valuable resources that the body could then use for something. The only other thing to mention here is something that you may come up in a suggest question. It's about fatty livers. Fatty liver is just basically where this NAD is, because it's being used up so much to help try and break down the alcohol, it can no longer oxidize fats. So therefore I get these fatty acid buildups within the liver. But it's not in the specification, it's just to help you with your further knowledge around alcohol. So that's everything we need to know about the functions of the liver. Guys, I wish you good luck in your exam. Don't use the words the amount and size, and good luck. All the best.